Welcome everyone to this, which is the second webinar run by Whitehall Training, and this is an introduction to good laboratory practice. For those of you who don't know Whitehall Training, we're the training business of a company called Infonetica, which is a UK-based software company with worldwide customers. We focus on products for the life sciences sector, especially CTMS, which is clinical trial management systems, for those who don't know, ethics review systems and tissue tracking software. We've got customers from universities and hospitals, contract research companies, pharmaceutical companies and logistics companies. So quite wide range, but mainly focused in the life sciences sector. As far as training is concerned, we've got a wide range of online courses written by expert authors, and that includes our speaker today, who is Roger Chapman. Roger is an internationally recognized expert in the interpretation and application of good laboratory practice to all types of regulatory studies, and that's both lab studies and field studies. He's also got extensive experience of good clinical practice in the laboratory, which is also known as GCLP, as well as good manufacturing practice in quality control lab environment. So before I pass you over to Roger, I'd like to open a quick poll for everybody, and this is really just to find out sort of what your level of expertise is already, what kind of sector you're in, etc. When I open the poll, if you can't see it, then you may have, I'll just open the poll now. You may have a little box on the top right of your screen with 3 squares. If you click that, you can open a view where you can see panels, and you ought to be able to see the poll that should let you fill it in. So it's just 2 questions, so shouldn't take long, but I'll just give you a few minutes to fill it out, and then I will share the results with everybody. Okay, so as I said, this is just really to get an idea of people's existing knowledge, so Roger knows how to, sort of what level to pitch things. The other thing is that if you have any questions whilst the webinar is going on that you'd like Roger to address at the end, just put them in the chat box to Roger, and then as long as we've got sufficient time, Roger will address them at the end. If there are any that we don't have time to address, then what we'll do is we'll have your email address, so we will email you directly with the answer. So just use the chat function to pass any questions to us that you want. Okay, well, you should be able to see the results now. So we've got a fair number of people in pharmaceutical development, nobody in the agrochemical sector, some in specialist contract research, some in full service contract research, and some in academia. So quite a good split, but the majority in pharmaceutical development. And in terms of those of you who've used Whitehall, a little under half of you have used Whitehall, and then the rest either not used Whitehall or aren't sure. Okay, well, thanks very much, and I'll just pass it over to Roger.
Thanks, Ashley. Thank you very much indeed for joining this afternoon's webinar, everyone, and welcome to this Whitehall training session about good laboratory practice. Now, as Ashley very kindly already said, I've been in the industry primarily around GLP for about 100 years now, so there's no way I can cover everything to do with GLP in 45 minutes. So this is necessarily just a very high level overview of the quality system. So what is GLP? What is good laboratory practice? So the definition of GLP I've stolen here from the OECD. The OECD is the Organization for Economic Cooperation and Development. They're a global body that deals with all kinds of things, but it includes GLP, quite a fortuitous, I guess, position for GLP to be finding itself in. But the OECD defined GLP as a quality system to do with the organisational processes under which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, archived and reported. So you can see there from that definition that it's a quality system that covers the whole conduct of non-clinical studies, from early concept through planning through the final delivery, and then the retention of all the records to support that study at the end. So it's what we call a real cradle to grave quality system. And the studies that we're talking about, GLP studies, non-clinical health and safety studies, are studies which are required by regulatory agencies to provide data to enable the hazard risk assessment process to be done by those regulatory bodies. And they're really designed to protect consumers, mankind, and the environment before new products are released for general use onto the marketplace. So what's the real purpose of GLP? What's it for? Okay, the overriding principle behind good laboratory practice is to promote the quality of test data. So it's all about the data. And you'll hear this come up again as we go through the presentation, but the primary focus of GLP is around quality of test data. Now, the concept behind GLP is that if a study is conducted in accordance with the principles of GLP, then the data that come from that study will be of robust and reliable quality. And what that means is that those data can then be used by various regulatory bodies around the world. So if the studies are done properly the first time, they don't then have to be repeated to satisfy the regulatory requirements of various agencies around the world. So basically, if the study is done once and it meets GLP requirements, then those data are accepted all around the world pretty much in terms of the risk assessment process as part of the regulatory pathway. In terms of the scope of GLP, as I've already said, it's non-clinical testing. Sometimes people will use the phrase preclinical, but the pace of regulatory development these days is accelerated so much that often the non-clinical and the clinical phases will overlap somewhat. And as we've already said, the scope includes both pharmaceutical products under laboratory conditions, but also environmental aspects of things like agrochemicals and industrial chemicals that can find their way by accident or by design into the environment. But I think there were no delegates online today that was particularly relevant to. And as I've already said, again, safety is the key in terms of the scope of the products. So very much within scope are pharmaceutical products, new drugs, whether they're biologics or old-fashioned small molecule products, pesticides. Some people are quite surprised very often to see cosmetics on the list, particularly here in the UK. We don't test cosmetics in this way anymore, but there are still some countries around that will test cosmetics in the laboratory for safety, or more particularly, I think, components of cosmetics get tested. It includes vet drugs, food additives, so that's human food additives, artificial sweeteners, for example, feed additives. So those are things like antibiotics that could be added to animal feeds, industrial chemicals, and in some cases the safety aspects, but not the functionality of medical devices are tested under GLP conditions. The types of study we're talking about, broad spectrum toxicology, so everything through acute contact testing, acute lethality testing, all the way through to 2-year and sometimes longer carcinogenicity tests, mutagenicity studies, so studies looking for genetic mutation, analytical chemistry, clinical chemistry, so bioanalysis is very much within the scope of GLP when it's applied to non-clinical study samples. Physicochemical properties testing, part of the specification, the characterization of products, falls under physchem properties testing. Some more environmental stuff, but as again I said, we don't appear to have any delegates that are so focused on that today. And that some of the ecotoxicological studies are really quite complex and quite far-reaching in terms of their design. So they're interesting, but I guess not for the folks today. And I've said this really by exception, but now quite explicitly, GLP does not apply to the analysis of human clinical trial samples. Okay, now that's not to say that people don't apply those principles anyway, but from a legal perspective, from a regulatory perspective, all aspects of a human clinical trial should be conducted under GCP or good clinical practice. So it's not a legal requirement to apply GLP to the assay of samples from clinical trials. Now, Ashley already mentioned GCLP, which you can probably imagine is a bit of a hybrid between GLP, good laboratory practice, and good clinical practice, GCP. That's an entirely different topic in its own right, but really what I'm doing here is just clarifying that it's not a legal requirement to apply GLP to clinical trial samples. This is just a quick look at the kind of classic diagram for drug development, so pharmaceutical product development, just to show you where GLP fits into the kind of broad spectrum of that pathway. Now, there's an awful lot of research work goes on upstream, optimization studies, screening studies will be conducted in various environments, but there's no current regulatory expectation for any kind of quality system to be applied to those. So they're technically known as non-GLP studies, and they're largely conducted upstream. So by the time that product pipeline is filtered down to studies that require GLP, we're often looking at a fairly significantly reduced portfolio of molecules or products. And from GLP onwards, the number of products in the pipeline just diminishes quite significantly. So by the time we get to clinical trials with humans, maybe 1 in 10 or even less of the products that have been through GLP will be determined to be safe enough to reach the clinical trial stage. So we'll do a very quick trip down the history road to look at the background of GLP. Like so many regulations that we live our lives by today, GLP was really born out of disaster. And you could say that the wheels very much came off the testing processes that were in place when it became necessary to implement GLP. Now, what happened was, back in the 1970s, the early to mid 1970s, there was quite an established testing program in place around the world, quite a significant volume of laboratories up and running in the United States performing safety tests on all the products we've talked about, some government labs, some commercial labs. And until that time, the US government, particularly the FDA, the Food and Drug Administration, and the Environmental Protection Agency, the EPA, had generally considered that all the data that they were being asked to include in their risk assessment processes were perfectly valid, reliable results. But over a period of time, those agencies started to become aware of quite a lot of feedback, query, concern from the general public in the United States particularly, that were raising questions about the real safety of products. So, for example, people were concerned that their kids had walked through a freshly sprayed crop in farmland and it had become ill or developed a rash, or people had been taking drinking soft drinks, for example, with artificial sweeteners in, and were getting adverse reactions to those kinds of things. So what I've always described as newspaper type headline feedback was reaching the agencies, and they began to realize that actually there might be something that was worth investigating. So what they did was they put a group of experts together from the government agencies and sent them out into the testing industry to see what was really going on. Now, what that survey process found was quite a wide range of issues in the established testing industry in the United States, and really quite widespread failure to perform reliable work. And I'll just give you some examples here. We'll run through these without a huge amount of detail, but individual studies were being run according to a plan, or at least a plan had been produced, but then people either didn't bother to read the plan or they did what they thought was best anyway. So there were lots of departures from the plan for the original scientific design of the study. The inspectors found people were performing tasks and roles that they hadn't been properly trained for. They'd been asked to do something new without being shown how to do it. Quite a lot of issues really with laboratory animals being not looked after correctly, so they were not necessarily always in the best of health. And of course, if you're working with a biological test system like rodents, for example, and they're not healthy, then their biological systems internally won't be responding as they would normally do. So you're likely to get flawed results from your tests. In the worst cases, they found that mouse studies were being housed in cages designed to hold rats. So the bars on the cages were too far apart. The mice could very easily get out and back in again. And in the very worst cases, mice were breeding in other cages and then returning to their own cage. So there was quite a lot of crossover from one treatment group to another. And in some cases, studies were ending up with more animals than they started with. So the control around the biological variables were quite horrendous. Animals were drowning in malfunctioning watering equipment. At the technical level, a lot of these studies were being exposed to the test compound by mixing the new chemical in with feed for the animals. But on some occasions, the compound wasn't being mixed correctly. So the rodents were simply eating the diet and leaving the chemical in the feed tin. And then, of course, they're not being exposed to the chemical at all. So the study design is fundamentally flawed. A few instances where animals had died, either through being drowned in the watering system, or had escaped and become lost, or had died as a result of treatment, and the technicians thought it was acceptable to simply remove those dead animals from the study and replace them with a new animal. Now, whilst that keeps the numbers up, of course, what it means is the new animals have not been exposed to the chemical for the same period of time as their cage mates. So again, the results are just completely flawed and unreliable. And then we begin to get into some examples of fraudulent behavior, fraudulent practices on part of the technicians and the management in the organizations. So if tasks had been forgotten to be conducted, then staff would simply make up the results. And in some cases, management were fabricating whole sections of data just to complete a report on time without actually conducting the work at all. So quite a horrendous range of examples there from the mid 1970s and across a number of laboratories. Now, you'll see the name for one particular company there, Industrial Biotest. They were the worst of the worst. And at their peak, they were a huge organization conducting almost 60% of all toxicology type tests in the United States. So you can understand that the data coming from that lab is now, the quality of that data is no longer believed by anybody when they've seen all of these examples of bad practice. The outcome was that IBT was closed down, senior staff were sent to jail, and a number of other laboratories were also subject to legal proceedings. But the real outcome as far as this presentation goes is that, of course, the government had to do something about this to improve the quality of the work that was being conducted. And so in a nutshell, GLP was formulated. Okay, so now we can kind of fast forward to today, just whiz through the calendar. So what the FDA did, the Food and Drug Administration, was published GLP in the late 70s, became law in the United States halfway through 1979. And that almost matches exactly the day I started in this industry. So I've kind of grown up with this. And as you'll see from this timeline here, very rapidly since the promulgation of GLP in the United States in the late 1970s, a number of other countries, really led by the OECD, have followed suit. It's interesting, if you scan down the list, that GLP was law in the United States in 1979, but only became law in the European Union and indeed the UK here from the late 80s onwards. So quite a big gap before it became a legal requirement elsewhere. Anyway, that's enough of all that kind of historic stuff. What I'm going to do now is just run through the section headings in GLP as they appear today and give you a little bit of explanation about the various components that are required. So GLP, as I've already said, begins with planning, talks about organisation and personnel, requires that management are appointed within a facility to take responsibility for the whole quality system. Introduces the concept of the study director. The study director is an individual who is appointed to each GLP study, and they have absolute responsibility for the quality and integrity and compliance of that study. So that's a very significant role. GLP has evolved a little bit in the last 40 years or so, and now allows the concept of what's called a principal investigator, which is really like an acknowledgement that the study director can't be in more than one place at one time. So with the current technology and ability for communication across various companies, we quite commonly find that GLP studies are run across more than one location. So the animals, for example, may be in one facility, but the analytical processes may be getting conducted in a different facility, possibly even a different company. So GLP, whilst it retains the single point of control for the study director, allows local principal investigators to take responsibility. Talks about responsibilities for individual study staff involved. Again, introduces a new concept here called quality assurance. Gives you a little bit of description around the facilities, what their design should consider. Some guidance on the stuff that we use in GLP studies, so apparatus, materials, reagents. Talks about test systems. Now those could be animals, they could be bacterial cultures, they could be analytical systems. What I'm going to do is just expand on these as we go through the rest of the presentation. Test and reference items, gives you some definitions around those. Talks about standard operating procedures. Those are very simply documents that describe how procedures are operated in a standard way. And what they're trying to do is to reduce the variability and increase the reliability of the conduct of studies by, as I said, minimizing as many variables as possible. A section on actually performing studies and right through to reporting study results at the end of every GLP study. And then a section on retaining records and specimens and samples that are generated during the conduct of the study. Okay, so what I'm going to do is go through those sections, just really one slide at a time, just to give you a little bit more flavour of what each of those requirements are. So management is a small group of people. It can be as small as one person. In bigger organisations, it can be a team of 10, 20, 30 people maybe. If you've got 1,000 people in a GLP organisation, then you'll need a team of management individuals to control all of those folks. So, but they have very specific responsibilities to ensure the necessary facilities, materials, equipment and so forth are in place for the duration of each study. So management have to be involved in the planning process, to make sure that whenever they start a GLP study, everything that's required to maintain that study through its lifespan will be ready and available throughout that process, because it's not sensible to have to suspend a study or postpone a study because you're running out of equipment or supplies. We've already talked about the study director, the single point of control, the conductor, if you like, of an individual GLP study, and how that if space-time travel is not possible, then they'll need some help on a local geographical basis for delegated phases to other facilities. As I've already noted, that the study director is a single point of control. So for every GLP study, there is one individual appointed as study director. GLP does not really allow those responsibilities to be shared, because in the investigations that the FDA and the EPA were conducting back in the mid-70s, what they found was that the more people that were involved at the kind of decision-making level, the poorer those decisions became, and the more likely it was for an individual to leave a decision to somebody else. So the concept of the single point of control is fundamental to GLP. Another fundamental concept is planning has to be absolutely in place and very well thought through. So a GLP study cannot start until the plan for that study is approved by the study director. And of course, distribution of the plan is important, because if you don't have the plan, you won't know what's in it. So if people are involved or expect to be involved in studies, then they must have access to the study plan. Staff have to be trained in their relevant procedures, and they're specifically responsible for the quality of their own data. Now, GLP is, I've already said, all about the data. So every step along the way, there is an expectation to record evidence that these requirements are being met. So we'll see documentation that the study director has been appointed. We'll see documentation the plan has been approved. We see documentation that people have been trained and so forth. So throughout the conduct of every GLP study, there's a constant burden for recording data and evidence. Quality assurance has to be appointed by management, and they're slightly different from QA that you might find in GCP, good clinical practice, and certainly different from GLP in manufacturing practice, in GMP, because in GLP, QA is very independent of the conduct of studies. So QA's role is to go monitor studies, to go perform inspections, to assure management that GLP is in place, and also to assure that the study plan is in place and is being followed. And the same with SOPs, standard operating procedures, just to make sure that they're really there and the staff are familiar with them and everything is being conducted in the way that it was intended to be. The GLPs require QA to do 3 types of inspections. So study-specific inspections, that involves reviewing the plan before the study commences, and also turning up in real time in the laboratory or wherever processes are being conducted in the facility, to really first-hand observe the conduct of procedures that are relevant to a particular study. At the end of the study, QA also has to audit the draft report to make sure it's complete, honest, accurate, the truth, the whole truth, and nothing but the truth. Now, in big facilities where there's a huge volume of studies and constant repetition of procedures going on, GLP recognizes that it's not practical to try and watch everything on every day on every study. So they allow really a sample-based approach to the observation of procedures, and those are called process-based activities. They're quite common in the bigger laboratories. And GLP also requires kind of a helicopter view of the whole GLP quality system. So that's done on a facility basis, where QA will look at the whole program to support GLP compliance. So that's a much wider view than specific studies or specific processes. In terms of facilities, well, GLP is not going to tell you what materials to use to build your facility or how to design your laboratory layout, but it just gives you some things to think about in terms of process flow, minimizing the risk of disturbance, minimizing the risk of mix-up or cross-contamination. And there's a general expectation that there's adequate space to do the job in a calm and controlled manner. So very cluttered laboratories, for example, just increase the risk of people mixing up samples, mixing up records, losing records. So GLP facilities tend to be quite neat and tidy and well organized. Test systems, those are the animals or bugs or cell cultures or whatever it is that we're testing on, must be looked after appropriately. Now, there's such a wide range of test systems these days that GLP can't possibly specify exactly all the conditions that are needed for every species or every type of culture. So there's very general guidelines here. Similarly, with test and reference items, what GLP requires is that the materials are adequately characterized, identified, and most importantly, separated from one another so that we can be sure we're only testing one item at a time. Some requirements around archives in terms that they should prevent the untimely deterioration or loss of stored records and samples. So apparatus, materials, reagents. The kind of catch line with GLP is that everything should be fit for purpose. So all apparatus, whether it's measuring equipment or mixing equipment or storage facilities or computerized systems, they should be qualified to the extent where management are happy that those apparatus are fit for their intended purpose, whatever that is. So GLP talks about systems also that control environmental factors. Now you can understand that that's important in a fridge or a freezer, for example, or a cold storage room, but also when we're working in laboratories, some reagents have to be kept below certain temperatures. And of course, in animal facilities, then the environmental conditions that maintain the health of those animals are just absolutely critical. So GLP expects that there's knowledge of fit for purposeness of your equipment, which involves periodic inspection, cleaning where necessary, routine preventive maintenance or reactive maintenance where necessary. And where a piece of equipment is being used to capture a measurement, and that measurement is important to the outcome of the study, then there's an expectation for calibration, even to national or international standards. Test systems, whether they're biological, chemical, physical, they need to be fit for their purpose, well characterized, and well maintained. And that includes apparatus used to generate physicochemical property data. So make sure that any balances or analytical instruments are of appropriate design and capacity. Toxicology studies were the kind of birthplace of GLP, as we know, but GLP now covers a wide range of study types. But I think it's absolutely critical that we remember that the animals are precious components of biological test systems, and therefore they need to be looked after as well as they possibly can. And quite commonly now in GLP, we see a kind of hierarchy of regulatory requirements, and almost without exception, any regulatory restrictions that are placed on the use of animals will supersede the requirements of GLP. So if an animal is seen to be suffering, then normally that suffering is terminated, even at the expense of the outcome of the GLP study. So we have to undergo ethical approvals to use animals in GLP studies, and I think that's absolutely right that that's the way it is. Test and reference items, we need to know as much as we possibly can about the product or the material under test. And the reason for that is at the end of every study, the study director has to draw a conclusion about the outcome of the study. And of course, if you didn't know what you tested, then you can't really do that. So we have to know as much information as we possibly can about the material before we begin. Everything has to be accounted for. So every weighing action, for example, is the weighed in, weighed out. There's an accountability process just to make sure that there's evidence that the study was dosed or administered in the correct way, and the dose levels required by the plan have been properly followed. Storage and handling mechanisms should minimize the risk of contamination or mix-up. We talked about test and reference items. Test items obviously are the unknown, which you're trying to find out more information about. Occasionally, but by no means every time, we also use a reference item. So that could be a marketed product, it could be a known agent that elicits a positive response for comparative purposes, or sometimes to validate individual tests. SOPs, standard operating procedures, they're simply documented procedures that describe how we do things around here. So they're specific to an individual facility, to an individual laboratory. They're authorized by management, and they really form the roadmap for the conduct of all the procedures in a facility. Their purpose is really to promote the quality and the integrity of data by minimizing as many variables as possible, and particularly the variability that's inherent from human operators. Quite often used as a training guide, so people will be trained against the SOPs by using the SOPs as a training manual almost. Now, I've already said that at the end of a study, we keep all the records from that study. We also keep historic SOPs. SOPs can evolve over time, they can constantly be improved. So to know how we performed a particular procedure last year, 5 years, 10 years ago, we'll need to refer back to historic versions of SOPs. So we have to keep all those as well. SOPs are documents that are version controlled, so the staff know when they've got the current version. I've shown a paper binder there, but these days they're mostly distributed electronically. So on to the performance of the study. I've already said, I think study plans are specific to individual studies and they must be authorized before the study can begin. If work begins on a study which is intended to be a GLP study before that plan is signed, the study is quite simply out of compliance. So it's a real black and white go, no go point in the process. GLP lays down a whole list of things that have to be in the study plan. We need clear objectives. Why are we doing this study? What do we hope to achieve? What are we testing? What are we testing it on? And the various experimental steps and processes that are being planned through the conduct of the study need to be mapped out in the study plan. Some people will still call study plans protocols. That was the original GLP language, but that's largely been superseded by the term study plan these days. Now, I've already said that everything that happens gets written down. Some of you may also have heard the phrase, if it ain't written down, it never happened. Well, that's not strictly true, but what is true is if it isn't written down or the record is lost, then we don't have that evidence anymore. So I think it's important to understand that GLP does bring a significant burden on the operators to record everything that's performed and all the weights and measurements that are taken and observations that are made and so forth. And those records need to be traceable to the person that made the record in the first place. So we live with the expectation that everything is signed and dated or initialed and dated, certainly on paper. With computerized systems, then the identity of the operator has to be captured by the system itself. GLP allows you to make changes to records in a timely manner if you correct minor discrepancies or errors, but also forces us to write an audit trail so there's a clear explanation of any changes. Data integrity, we don't have time to talk about that in great detail today, but it just underpins everything that GLP talks about, because the validity of data, as I've already said, is absolutely pivotal. So nearly at the end of the study, we have to produce a report. The director has to produce a report. You cannot escape that. It's a fundamental requirement of GLP. It's a fairly standardized document construction that talks about the methods used, the material that was under test, the results obtained, and where appropriate, a conclusion that's drawn from the results that have been obtained. Now, I've already said when we were talking about QA that QA has to audit the draft report to make sure it's complete, accurate, honest. And the kind of expectation here is that QA will verify that there's everything that happened in the study is reflected in the report, and vice versa, that there's nothing in the report that didn't happen in the study. So it's a kind of a two-way audit process, but it's a fundamental requirement of GLP that QA do that, and then they will finally sign the report to make sure that it's clear that that audit has been satisfactorily completed. And the study director, or in the case of a phase report, the principal investigator, also sign a statement to talk about the level of compliance with GLP that was evident throughout the study. GLP is now getting quite tight on the requirement to produce a summary report, even for aborted or partway through cancelled studies, just so that there's no suspicion or no possibility that adverse results are being hidden from the regulatory gaze. So even if a study produces adverse results, then there's still an expectation for a report. And finally, I think we're at the point where the report is finalized, it's all signed off, and then the study director has to insert all the materials into the archive for safekeeping for a significant period. If there are histopathological specimens, for example, or a sample of the material that was tested, the test item, those are also retained in the archive. And everything that supports the conduct of the study, so the plan, the report, QA records, facility records, they're all retained in the archive under tight access control conditions. GCP and GMP, good clinical practice and good manufacturing practice, basically just say keep stuff, okay? But GLP has a whole section on the access permissions that are required to get materials in the archive, the kind of levels of protection that are required. So it's quite a specific set of expectations in GLP for the retention of records. It's very difficult to answer the question, how long should we keep stuff for? Because different regulatory agencies have different timeframes. So in practice, what happens in a GLP archive is the archivist just contacts the sponsor, the people that commissioned the study at intervals, and say, I've still got your stuff. You still want me to keep it, or do you want it back by now? So the GLP organizations often won't drive the full retention period. That's down to the sponsor who commissioned the study in the first place. And just one very brief slide to show you how GLP has had to react to the restrictions that COVID-19 has placed upon us all. Government inspections of GLP facilities, so agency inspections from the MHRA, for example, here in the UK, or the FDA in the US. A lot of those inspections are now being conducted remotely, so by telephone call or video conferencing. I haven't heard any adverse feedback about that process. It seems to be working okay. There was specific guidance from the MHRA on the impact of any delayed or missed internal QA inspections. Remember, I said we have to do 3 types of QA inspection. So if those are getting delayed because staff can't get in, or the studies are being delayed, etc., then there has to be a risk assessment to determine the impact of that. Similarly, if maintenance engineers can't get to you to calibrate balancers or repair your HPLC or mass spec or whatever it is, then there also has to be a risk assessment for that. And also some quite specific guidance has been issued on approving documents remotely. So suddenly the whole world is working from home, but documents like study plans, SOPs, for example, still need to be formally authorized. So there's been some guidance around that, and the simple solution is print, sign, scan, and send back, and that's been perfectly acceptable. So just to wrap up and to remind you that, you know, the overall objective of GLP is to promote the quality and validity of the data. It's all about the data. And increasingly, it becomes important to be able to reconstruct studies, sometimes many years later. And it never ceases to amaze me sometimes how long the overall regulatory process is, so that many years can go by before an early GLP study gets submitted to the authorities. And if for any reason they decide they want to have a look at the validity of that study, then we have to pull data from the archive that can be over 10 years old. And of course, if you're trying to understand what happened 10 years ago, the chances are that the study director will have left, and everybody else that was involved in the study will also have left or forgotten. So the only thing that's left at that point is the data. So your studies really are only as good as the data. Okay, just a couple of slides of bedtime reading, really. I'm not going to go through these, but just to show you the volume of further guidance that's been released by the OECD. If you just put OECD GLP into a search engine, you will find all of these documents. They're all freely downloadable, many in multiple languages. You can see from the titles that they're quite specific guidance documents, but they're constantly being added to. Some more stuff from the OECD. Very helpfully, they produce frequently asked questions. I don't think they're truthfully that frequently asked, but they're things they want you to know. So again, a range of topics there and a web address. But if you just Google OECD GLP FAQs, you'll find all of these. Some more general questions and answers get published from time to time, and some good explanation of what the mutual acceptance of data means and how that works. So you remember right at the start, I said that if you run a GLP study in one location, those data will be accepted elsewhere. And that's what the mutual acceptance of data is all about. Okay, so that's me done. I'm quite happy to try and answer some questions via the chat function now, if you have any, or by all means email in, and I'll try and address those for you later. So if there are any questions on the chat function, please ping those in now. Could I comment on what GCLP is? I'm just catching that as it ghosted up on my screen. Yeah, GCLP is good clinical laboratory practice, and that's the kind of hybrid really between all the stuff we've talked about for GLP now, and how that works for clinical samples. Now, in the early days of analysing clinical samples in laboratories, people either applied no quality system at all, or they applied GLP. Now, it very quickly became realised that there was a lot of requirements in GCP, in good clinical practice, that are specifically designed to protect the rights and welfare of the patient, the subject, and GLP doesn't address those at all. GLP is all about the data, and if I could be brief about it, GCP is primarily about the rights and welfare of the subject, the human subject. So there were some gaps between the two systems. So GCLP looks like GLP, but it addresses things like ethical approval of the analysis of samples. It looks at the consent of the subject to have their samples analysed. It looks at things like what to do if you get adverse results that need to be immediately reported back up the chain to the clinic. So it really fills the gaps between GCP and GLP, and it's a separate subject in its own right. I think Whitehall actually have some online training specifically to cover this, but it is a different topic. If you want to know much more about it, then I'd recommend you pop an email question into me and we'll try and address that in a bit more detail. Were there any other questions? Okay. So I'm just going to hand back to Ashley now to wrap this up.
Thank you very much, Roger. That's great. Just thank you all for listening. I just wanted to tell you about our next webinar that's due in 2 weeks. We'll be covering good pharmacovigilance practice. For information and to register, you can visit whitehalltraining.com/pharmacovigilance-webinar. I will be emailing everyone with a copy of that link and a recording of this webinar, so you don't need to take a note of it now. For those who don't know, pharmacovigilance is the process by which side effects and adverse event reactions to drugs are captured, communicated to medical professionals and patients. Now, it takes place before, during, and after drug trials. And depending on when it happens, the mechanics and the challenges are quite different. So we'll be introducing all of it. We'll be introducing how pharmacovigilance works during a drug trial, but also in post-marketed drugs. So thanks again for attending this webinar. We have got a more detailed GLP course for those who are interested, again written by Roger, so obviously covers a lot more detail than we could do in just this webinar. So you can get information by going to whitehalltraining.com. I will be emailing everyone a special code so that if you want to personally take the GLP training, you can use it at checkout to save 50%. If you're interested in larger orders, so if you're ordering for a number of colleagues, then email me directly at ashleysmith@infonetica.net and I can give you a price. Also email me if you want to be invoiced. So if you want more in-depth information about GLP, you have specific questions, you'd like to discuss bespoke training, or you're interested in GLP audits, then please contact Roger at his email address, roger@chapmanqa.com, or you can visit his website there, so www.chapmanqa.com. So just thank you again for all of you attending, and I will be in touch soon. Thanks. Bye.